Hi guys, today I will be discussing ultrastructure of a bacteria. Bacteria possess a cell wall, cytoplasmic membrane, nucleoid, ribosomes, and occasionally inclusion granules. Also, structures which are external to the cell wall. Cell wall. The bacterial cell wall is an extremely important structure, being essential for the maintenance of the shape and integrity of the bacterial cell. The primary function of the cell wall is to withstand the osmotic pressures. Most bacterial cell walls have in a common unique structural component called peptidoglycan, also called murine or glycopeptides. Exceptions include the mycoplasmas, extreme halophils, and the archaea. Chemically, the cell wall is composed of peptidoglycan. Peptidoglycan is formed by N-acetylglucosamine and N-acetylmuramic acid alternating in chains, cross-linked by peptide chains. Gram-positive cell wall. The cell walls of gram-positive bacteria are quite thick and consist of between 60% and 80% peptidoglycan. Embedded in it are polyalcohol called teichoic acids. Some are linked to lipids and called lipoteichoic acid. Lipoteichoic acid link peptidoglycan to cytoplasmic membrane. The functions of teichoic acid are, gives negative charge, transport ions, anchoring, external permeability barrier. Gram-negative cell walls. Gram-negative bacteria contains less peptidoglycan. A second membrane structure is found outside the peptidoglycan layer. Outer membrane is found only in gram-negative bacteria. It functions as an initial barrier to the environment and, it is composed of lipopolysaccharide and phospholipids. Lipopolysaccharide. The LPS present on the cell walls of gram-negative bacteria account for their endotoxic activity and antigen specificity. A bacterium is referred as a protoplast, when it is without cell wall. Cell wall may be lost due to the action of lysozyme enzyme, which destroys peptidoglycan. A bacterium with a damaged cell wall is referred as spheroplasts. It is caused by the action of toxic chemical or an antibiotic. Cytoplasmic membrane. The cytoplasmic membrane is a phospholipid bilayer, with proteins distributed randomly throughout. A major difference in chemical composition between prokaryotic and eukaryotic cells is that, eukaryotes have sterols in their membranes, example cholesterol, whereas prokaryotes do not. Functions of cytoplasmic membrane: transport of nutrients, energy generation, electron transport. It is the location for regulatory proteins and biosynthetic proteins. It acts as a semi-permeable barrier between the cytoplasm and the cell environment. Mesosomes. They are vesicular, convoluted tubules formed by invagination of cytoplasmic membrane into the cytoplasm. They are principal sites of respiratory enzymes and help with cell reproduction. Cytoplasm. Cytoplasm is about 80% water, and contains primarily proteins which are enzymes, carbohydrates, lipids, inorganic ions, and low molecular weight compounds. Cytoplasm is thick, aqueous, semi-transparent, and elastic. The major structures in the cytoplasm of prokaryotes are a nucleoid, containing DNA, particles called ribosomes, and reserve deposits called inclusions. The nucleoid. The nucleoid of a bacterial cell usually contains a single long, continuous, and frequently circularly arranged thread of double-stranded DNA called the bacterial chromosome. This is the cell's genetic information, which carries all the information required for the cell's structures and functions. The enzymes topoisomerases control topological changes in DNA. The nucleoid can be spherical, elongated, or dumbbell-shaped. In addition to the bacterial chromosome, bacteria often contain small, usually circular, double-stranded DNA molecules called plasmids. These molecules are extrachromosomal genetic elements. That is, they are not connected to the main bacterial chromosome, and they replicate independently of chromosomal DNA. Plasmids may carry genes for activities such as antibiotic resistance, tolerance to toxic metals, the production of toxins, and the synthesis of enzymes. Ribosomes. The cytoplasm is densely packed with ribosomes. They are smaller than eukaryotic ribosomes. The endoplasmic reticulum is not a component of prokaryotic cells. Bacterial ribosomes are 70s in size, made up of two subunits of 30s and 50s. Inclusions. Within the cytoplasm of prokaryotic cells are several kinds of reserve deposits, known as inclusions. Cells may accumulate certain nutrients when they are plentiful, and use them when the environment is deficient. Endospores. When essential nutrients are depleted, certain gram-positive bacteria form specialized resting cells, called endospores. Endospores are dehydrated cells, with thick walls and additional layers. They are formed internal to the bacterial cell membrane. Now, let's study the structures external to the cell wall. GLYCOCALYX. GLYCOCALYX means sugar coat. GLYCOCALYX is the general term used for substances that surround cells. The bacterial glycocalyx is a sticky, gelatinous polymer that is external to the cell wall and composed of polysaccharide, polypeptide, or both. If the substance is unorganized and only loosely attached to the cell wall, the glycocalyx is described as a slime layer. Flagella. Some prokaryotic cells have flagella. They are long filamentous appendages that propel bacteria. Function of flagella is motility. For MCQ, bacteria that lack flagella are referred to as atrichous. The arrangement of flagella may be described as monotrichous, single flagella on one side. Lophotrichous, tuft of flagella on one side. Amphitrichous, single or tuft on both sides. Peritrichous, surrounded by lateral flagella. Fimbriae and pili. Many gram-negative bacteria contain hair-like appendages that are shorter, straighter, and thinner than flagella. These structures consist of a protein called pilin. They are divided into two types, fimbriae and pili. Fimbriae. Fimbriae can occur at the poles of the bacterial cell or can be evenly distributed over the entire surface of the cell. Function. Adherence to surfaces like rock, adherence to epithelial surface in our body, formation of biofilms. Pili. Pili are usually longer than fimbriae and number only 1 or 2 per cell. Pili are involved in motility and DNA transfer. Thank you. Subscribe for more videos.